The overall goal of this methodology is to show how to measure the rheological properties within the microdomains of heterogeneous flow cell and static biofilms.
This method can help answer key questions on rheology in biofilm field, such as how specific matrix component, different species, or compound affect biofilm rheology and thus its response to physical force such as shear.
This is a non-destructive technique with high spatial resolution, and it can be conducted with equipment standard to biological laboratories.
After preparing overnight bacterial cultures and supplies and medium for flow cell setup according to the text protocol, aliquot fluorescent microspheres in sterile water into microcentrifuge tubes. To remove the sodium azide, spin down the tubes for 5 minutes. Under a sterile environment such as a flow hood, remove the supernatant and use 1 milliliter of growth medium to resuspend the microspheres. Then, add microspheres to the rest of the growth media for flow cell culture. Set up flow cell culture according to the text protocol. For a static culture setup for biofilms grown in microwell chambers, dilute overnight cultures 100-fold in microcentrifuge tubes by adding 10 microliters of overnight culture to 1 milliliter of growth medium containing microspheres. Then use the diluted cultures to fill the microwell chambers and incubate at 37 degrees Celsius under static conditions. On days 3 and 5,